zone and on a new plate. If no new colonies grow, then the bacteria in the zone are dead.
Thanks, Cassie.
Yeah. I guess reswab and grow out.
It didn't. Do
Okay, can
anything
somebody
near
confirm you
that.
can hear me?
I can.
Great.
You're gonna go over the notes pretty soon here, right, for the unknown project?
I'm sorry, can you repeat that?
You're gonna go over some of the notes for the unknown project, right?
No, I did that last time.
Oh, so you're not gonna talk about like when you're gonna send out some of the information for that?
Well, I'll mention that I'm going to send out some information probably tonight, probably very late tonight, meaning probably past midnight, but maybe around midnight. I'll send out the
Oh,
your first 3 test results.
so we shouldn't turn in anything as of now.
No, probably
There's
not.
There's nothing.
The only thing you
nothing,
can turn
yeah.
in is your number.
Yeah. Okay.
Just a minute, I'm trying to get there.
In like the text entry box, I'm assuming that's where I put it.
I'm sorry.
Oh, nothing. Never
I'm
mind.
not hearing you for some reason. Let me turn
Yeah,
up my speaker here.
my mic is a bit
There,
funky.
then I can hear you better. Say again.
Oh, I was just saying we put that in like the text entry box, all of our information as like bullet points. Kind of.
For your unknown project,
Yeah.
you're talking about the report or for the unknown notes pages?
Specifically the unknown notes pages, not the report.
Yeah, no, the notes can be done however you want. You
Sweet.
can put it in notes if you want, bullets. You can put it in paragraphs. You can put it in sentences. You can use phrases. I'm
Sweet.
not going to grade the grammar of your unknown notes page.
Awesome.
Okay. All right, I'm almost there. Let me call up the lab here. Hopefully it's lab 6. Let me look at that. All right, let me share my screen here. Got 2 Zoom sessions, so just a minute here. Getting a little confused. There we go. For some reason to share it, I always have to blow it up too. I can't do it when it's reduced in size. All right, you should be able to see my screen here. Today is August 8th. And we should be doing lab 7, throat and urine cultures. Let me remind you, you do have a quiz 3, this Wednesday, over lab 5, lab 6, chapter 4, and the end of chapter 3. I'm going to work on finishing grading chapter 6. Lab 6 after this lab. So that's my number 1 priority, so that you can have that to study by. I could mention some things. Are you guys seeing my screen? You should be.
Yeah, I am.
Okay. I should mention that I'm noticing that a number of students are turning in the same answers. And I don't mind you guys working together, but you should write the answers in your own words. And I know you're sharing answers, at least some of you, because you have the same answers and they're the same wrong answer. So it's pretty obvious to me that you guys are working together and one person probably came up with the answer and you're all sharing it. And that's not a good thing to do, because you're not learning when you're taking somebody else's answer. You won't be penalized in the lab for that, but I'm just telling you I'm aware of it. And like I said, when you get the wrong answer and you all get the same wrong answer, it's pretty obvious. There's even one student who thinks one student who thinks they're clever. And what she did was she put the order of the answers in a different order, but it's the same exact answers.
Before we get started, I have a quick question about the labs.
Yeah,
I know the labs are your priority over the resubmits, but with the
over the
resubmits,
what?
am I
Hold it,
over
I
the
didn't
res
catch what you said.
over the resubmitted, like when we return them back in for the
Uh-huh,
corrections?
uh-huh.
Um, to my knowledge, everyone here has not got graded for any of the resubmitted stuff. I just want to make sure that we're not supposed to like leave a comment in like any section or something, or you just haven't gotten to those yet.
I haven't gone back. Let me stop the share so I know what I'm talking about. I think I haven't, I think I have not graded anyone's lab for the second time, but I might have graded lab 00 for the second time. Let me look that up. No, that's lab 6. Let me go to the grade book. I have to not share the screen here because I'm showing everybody's grades because I don't even know how to answer that question unless I look at the grade book.
Yeah, I just didn't want to. I want to make sure I was doing everything right when I was resubmitting those. Didn't want to make sure you always
Oh
like
yeah,
miss any.
as long as you give the same format for your answer, I'm sure it's right. I think I got lab 00 graded a second time. Let me look that up. But other than that one, I don't think I've graded any others for a second time.
Okay.
It doesn't even show in the grade book because I've got lab 0 and 01 together. But I think lab 1, lab 00 was graded for the second time. Oh, good. My computer will speed up now. You're not seeing it, but it just popped up a window saying it finished grading not grading, finished with the Zoom lecture session. So now I can upload that page. I'm not going to upload it now, but I will do it later. But I've got the micro lecture for the lecture now. I don't know what type of a file it is beforehand, but it's not one that you can put on the internet and then run a video from. And it has to be converted into an MP4 file. And that's the file I can use to I'll get started on the second grading of the labs. As far as I'm aware, no one is submitting anything incorrectly. All you have to do is submit it again in the same format as you submitted it the first time. Because if you don't submit it the first time correctly, I will make a comment about it. You should be doing it this way. And usually people submit the worksheet. And if they do submit the lab module, it's usually only the first time. I've never had a student submit a worksheet the first time and then the lab module a second time. Obviously, if you did that, you would perhaps get a penalty. But beyond that, I can't see submitting it incorrectly. Just submit
Okay.
the answers.
Perfect.
Okay.
Yeah.
All right. Let's get out of the grade book, close that down, and then let me reshare my screen. Oh, this is the lab I'm grading. So fortunately, I don't have a student showing there. They're there. Don't want to show you that. Let's go to lab 7. That's the MP4 file. No, these are the lab files. Do I have them open? Yes. All right, so we're discussing lab 7, throat and urine cultures today. On Thursday, we'll have another lab 8. Don't need that. Don't need that. There we go. That's the exercises though. Let me move to the lab module. Throat and urine cultures, lab 7. There is some reading to do You can read about that. There will be a video clips of supplemental materials. The virtual lab no longer works for this lab. The primary learning objectives: 1, be able to explain how to properly collect throat and urine specimens for routine culture in the lab. 2, be able to list common bacterial pathogens isolated from throat and urine cultures. 3, know the culture media used to cultivate these pathogens, meaning the different media. And then 4, recognize which tests are used to identify throat and urine pathogens. 5, understand the principles and how to interpret the following tests: the catalase test, the coagulase test, and the oxidase test. We will talk about the XV test or 15-factor test, but you do not need to be able to explain that test. All right, once the specimen is received in the lab, they are logged and processed in a timely fashion. Specific culture media are inoculated and incubated depending on the specimen. And we're going to talk about specimens obtained from throat and urine specimens. Below is an overview of the culture media used to cultivate common bacterial pathogens isolated from the throat and urine cultures. For the throat, the main culture media is blood agar, and that's to grow Streptococcus pyogenes, which is the main human pathogen isolated from the throat. And if you don't know, Streptococcus pyogenes causes strep throat and a whole bunch of other diseases which have become fairly rare today because of antibiotics. And I think it was scarlet fever and rheumatoid fever and a bunch of other diseases, which I don't even know because I was born after the introduction of antibiotics, so I don't know any of these diseases. They used to be very common, but they are not common anymore, at least in the United States. We also have chocolate agar, which is a culture media used for the throat, and that's to grow Haemophilus influenzae, which is seen in children and infants. And those are the only 2 culture media and 2 pathogens you need to know about that are isolated from the throat. For urine specimens, we have 3 culture media. We always grow it on blood agar, and we also grow it on MacConkey's agar or EMB agar. EMB is eosine methylene blue. And we'll talk about why there are 3, and you always grow it on blood agar, as well as one of the other 2, either MacConkey's or EMB. MacConkey's and EMB are very similar to each other. And we'll talk a little bit more about that. Some of the common bacterial pathogens we see from a urine culture include Streptococcus species, Enterococcus species, and Staphylococcus species, and those are all gram-positives. They're also all cocci. And then there are 2 others, Enterobacteriaceae, such as E. coli, and Pseudomonaceae, such as Pseudomonas aeruginosa. These are gram-negative, and they're both bacilli. Any question about any of that? Throat cultures. Sore throats, if they have white patches on the inflamed tonsils or in the back of the throat, that is an indication of a bacterial infection. If the throat only looks red, that's likely a viral infection. The most commonly isolated bacterial pathogen in the throat is Streptococcus pyogenes, which is the name I use because it names the species, it is also called group A Streptococcus. That's an antigen distinction of streptococci, which was started, I think, by Dr. Lansfield in maybe the 1930s and 40s. And then it's also an abbreviation, GAS, for group A Streptococcus. I will always use the term Streptococcus pyogenes, but either term is correct. Strep throat is diagnosed usually in one of two ways. There will be a rapid test throat or throat culturing, where in the throat culturing, you swab the back of the throat or the tonsils where there is redness or white patches. And this picture, is trying to show you where to get it from the back of the throat or the tonsil. You do not get it from the sides of the mouth, the tongue, or the top of the mouth, which the top isn't shown there, but or from the teeth, which nobody would sample from the teeth anyways. Patients are then tested. If they test positive for patients that test positive for Streptococcus pyogenes are then placed on antibiotic therapy. This is because Streptococcus pyogenes is very difficult for the patient to throw off the infection. Streptococcus pyogenes is fastidious and it will not grow on a general purpose media. You have to use a media supplemented with 5% sheep's blood to facilitate its growth. And we call this blood agar plate, or BAP is the acronym. The plate is then examined for any small plates that have clear zones of complete hemolysis or beta hemolysis around them. And these could be Streptococcus pyogenes. We only need 2 biochemical tests to further prove that these colonies, which are showing beta hemolysis, are Streptococcus pyogenes. 2 biochemical tests. You do the Cadlase test and then the basotrace and susceptibility test. I can tell you in the story that when I was a child, they simply did the blood agar plate. And if you had complete hemolysis, beta hemolysis, they assumed you had Streptococcus pyogenes, which was not entirely correct. But I was a kid a long time ago, so. You use the Cadlase test where the bacteria are exposed to hydrogen peroxide. And if the bacteria can produce Cadlase, you'll switch the hydrogen peroxide to water and oxygen. The oxygen will create bubbling, and you look for the presence of bubbling. If the bacteria test positive for Cadlase, you see bubbles appear like this. If the bacteria are negative for Cadlase, then there will be no bubbling. Streptococcus species and Enterococcus species test negative for Cadlase production. So Streptococcus pyogenes will be negative in the Cadlase test. You then run the basotrace susceptibility test. Basotracin is an antibiotic, and what you do is you plate the bacteria from the beta hemolysis on the disc in this region here, and then apply the disc before the bacteria have grown. So you just plate the bacteria, this is not shown here, and then apply the disc about 5 minutes later, and then you incubate the plate. if the bacteria are sensitive to bacitracin, and there's a zone of inhibition, then it is Streptococcus pyogenes. If, on the other hand, there is no resistance to bacitracin, no sensitivity to bacitracin, then it is a beta hemolytic Streptococcus, but it is not Streptococcus pyogenes. So it's some other Streptococcus. Is that clear? So if it is sensitive to bacitracin, it is Streptococcus pyogenes. If it's not, it's another Streptococcus, and it's not Streptococcus pyogenes, but it is beta hemolytic because of that. All right, that's the main pathogen you have to find coming from the throat, and it's seen in children and adults, Streptococcus pyogenes. Let me mention that why this one is so difficult to throw off is that Streptococcus pyogenes produces a capsule, and that capsule prevents white blood cells from phagocytizing the bacteria. The capsule also protects the bacteria from the host immune system. Any question about any of that? And so children, before the advent of antibiotics, they would get a sore throat, and it would just last for a long time. And the bacteria would move from their throat and into their blood, and then could do a lot of damage to their body, including it could get, the bacteria could start to grow in their heart valves, and then later in life give them heart disease. I'm not sure what scarlet fever was, but the bacteria, once it could get into the blood, could also cause scarlet fever. And I don't remember what scarlet fever is because that was a disease that happened before I was born. And I just don't know, but you could look it up if you want. There's a couple of other diseases, I don't even know what they are, and they were all caused by Streptococcus pyogenes, causing an infection someplace in the blood after the bacteria invaded the blood, usually from the throat. Another organism that can be clinically significant when isolated from the throat is Haemophilus influenzae, a gram-negative coccobacillus. Haemophilus influenzae is not usually seen in adults and even older children, but it is the most common cause of ear, eye, and sinus infections in babies and in young children. Haemophilus influenzae is also fastidious, but it cannot lyse the red blood cells of a blood agar plate, so it will not grow on a blood agar plate. You have to grow Haemophilus influenzae on chocolate agar. Chocolate agar is the same as blood agar, except the blood is added when the agar is very hot, and that causes the blood to lyse, releasing the nutrients from the blood into the agar. And that also changes the color. Instead of a red color, the chocolate agar is brown, and that's why it's called chocolate agar. Haemophilus influenzae is a tiny gram-negative coccobacillus that forms tiny colonies. You don't need to know this, but you can get not chocolate agar. Brain is not working well today. You can get Haemophilus influenzae to grow on blood agar if you put a streak of Staphylococcus aureus down the blood auger, and then you streak Haemophilus influenzae perpendicular to the Staphylococcus aureus. What happens is Staphylococcus aureus will lyse the blood, but only in a zone around the streak. And then the Haemophilus influenzae, which is streaked out this way, will not grow in this region of the blood auger plate, but it will grow in the region around Staphylococcus aureus. And the reason why it grows right here is because the Staphylococcus aureus is lysing the blood, and then the nutrients from the blood are diffusing out, allowing the Haemophilus influenzae to grow in this region of the blood auger plate. Any questions about any of that? All right, the factors that Haemophilus influenzae requires to grow are the X and the V factors from blood. And that's all you need to know. X factor is hemin, a V factor is NAD. You don't need to know any of that, but Haemophilus influenzae does require something from blood, but it cannot lyse the blood. So by itself, it cannot grow on blood auger, but it can grow on chocolate auger. All right, any question about throat cultures? If not, let's move on to urine cultures. Urine cultures are used to diagnose urinary tract infections. Bacteria can travel from the urethra up to the bladder. And actually, if it does infect the bladder, it can go to the ureters and then can move up the ureters up to the kidneys. That's a very severe urinary tract infection. My mother actually once had that happen, and that was partly because she did not tell anyone that she was having symptoms of something, except for one caregiver, and this caregiver never told any family members. And for some reason, the caregiver just assumed that it was a woman's problem and it would go away, and it didn't go away, and it just got worse and worse. And what you want to do is once you have the bladder infected, go in, have yourself tested, and get treatment for urinary tract infection. It's much easier treating a urinary tract infection if it's only involving the bladder than if it's involving the ureters and the kidney. It's a serious disease if it gets into your kidneys. Bacteria typically travel in females from the urethra to the bladder because females have a shorter urethra than males. So females are more predisposed to developing a UTI than males. Any question about that? To sample for a urinary tract infection, the genital area is usually clean prior to collection. And then you do a clean catch midstream involving only collecting the middle portion of the urine. This is to reduce the cultivation of transient organisms from the urethra. The urethra does have bacteria growing on it, and most of them will be your normal microbiota, so it's not a problem. But if you do get a urine sample, you will get bacteria from your urethra. And so they try to minimize that by doing the clean catch midstream. Any questions about that? A better way to sample the urine is a catheter where you stick it up the patient's urethra into the bladder and then sample the urine directly. However, the catheter itself can take bacteria from the urethra and then put it into the bladder. So the catheter is not, what's the word I'm looking for, is not without problems itself. The best way to do it is to actually use a suprapubic aspiration, where you poke a needle into the bladder from outside and then sample the urine directly from the bladder. Patients, of course, do not want to have the suprapubic aspiration done. Who wants a poke of a needle? And patients aren't very pleased with the catheter either. So many times we still do, in fact, most times we still do clean catch midstream to get a sample of the urine from the bladder and then check for urinary tract infection. What you do is you splate 1/1000th, meaning 0.001 mil of urine placed on a selective media and blood agar plate with an inoculation loop. And this loop is designed so it takes exactly 0.001 milliliters of urine. And then you streak it on the blood agar plate, or you streak it on the McConkey's agar plate, and then that looks like they took the loop and then streaked it around. And like I said, with the urine cultures, we usually streak 2 plates. One is always blood agar, and that's to allow all organisms to grow, gram-negative and gram-positives, as well as fastidious organisms will grow on blood agar plate. And then you grow on either McConkey's or EMB. Both McConkey's and EMB select for gram-negatives. Why we grow on a McConkey or EMB agar is because most urinary tract infections are gram-negatives. Most are actually from the family Enterobacteriaceae, and the number one species which causes a urinary tract infection is actually E. coli. Any question about any of that? E. coli and gram-negatives will grow on MAC and on EMB, and then everything will grow on BAP, or essentially everything. Any questions about any of that? All right, I should mention that E. coli is a coliform. A coliform is just a microorganism that grows in your colon or your intestines. And there are many Enterobacteriaceae which can grow in your colon, and we call them all coliforms. The interpretation of a positive urine culture is multifaceted. Some factors that need to be considered is the type of collection. For example, voided versus catheterized specimen. The voided will have more contaminants than the catheterized specimen. And obviously the catheterized specimen will have some contaminants. The gender of the patient is important too. For some reason, males have fewer contaminants than females. The number of the colonies per mil is then assayed. The presence of polymicrobial growth is also looked at. If there are multiple organisms isolated, it may indicate a poor collection technique. Once you get a bacteria growing on the McConkey's and the blood agar plates. You then do testing to determine well, you have to isolate the species, grow it up as a pure culture, and then do testing to identify what the species is. One of the tests you do is coagulase. Coagulase is an enzyme that converts fibrinogen into fibrin and then can convert a plasma into a clot. So here we have plasma. It doesn't clump because coagulase is absent. If coagulase is around, it'll cause clumping. I will generally use the coagulase test tube test where you have the plasma and then you add the bacteria to it. If the bacteria produce coagulase, the plasma will clot. So this is coagulase positive. And if it doesn't, it'll not clot, and that'll be coagulase negative. Staphylococcus aureus is a bacteria which can cause a urinary tract infection, which is coagulase positive. And that's the main purpose for using the coagulase test, to help you distinguish Staphylococcus aureus from other species of Staphylococcus. Most other species of Staphylococcus are negative. Staphylococcus aureus is coagulase positive. But most commonly, bacteria isolate from the urine culture is E. coli. E. coli belongs to the family Enterobacteriaceae, but there are two families which can cause a urinary tract infection: Enterobacteriaceae and Pseudomonaceae. You can distinguish the Pseudomonaceae from the Enterobacteriaceae by the oxidase test. The Pseudomonaceae, particularly the human pathogen Pseudomonas aeruginosa, is oxidase positive. The family Enterobacteriaceae is oxidase negative. Remember, they're both gram-negative bacilli. Some other differences, the Pseudomonas are aerobic and they're non-glucose fermenting bacilli, gram-negative bacilli. The Enterobacteriaceae are facultative anaerobes, and they're all glucose fermenting. Let's talk a little bit more about the oxidase test. It's measuring an enzyme in the electron transport chain of aerobic respiration, cytochrome oxidase C. And I don't know why we don't call it the cytochrome oxidase C test. We just call it the oxidase test. It can separate the Pseudomonas from the Enterobacteriaceae. If the bacteria are oxidase positive, they will develop a purple color in 30 seconds. If they're oxidase negative, they will not develop the purple color. They will not change color. All right, any questions about the different tests we use to isolate the UTIs? All right, if not, let's move on to the exercises. You're not going to physically perform any of these activities. However, you will be responsible for knowing how to perform them. We have a little video for you to look on the Cadlase test. another video on the coagulase test, and a third video on the oxidase test. So watch those videos. I'm going to switch to the laboratory exercise now, and then answer the questions. Question 1, ask, what converts fibrinogen into fibrin? And you're using to detect the presence of catalase, coagulase, and oxidase for these questions. So you're talking about the enzyme that converts fibrinogen into fibrin. And then name an organism that tests positive for this enzyme. Sometimes people don't get 1B, but there's 1A and 1B. And then 2, name the enzyme that detects the presence of cytochrome C proteins in the electron transport system. So that's the, no, I guess I can't name it. That'll tell you what the enzyme is. All right, then 2, name 2 organisms that test positive for the enzyme. So 2B. And then 3A, name the enzyme that hydrolyzes hydrogen peroxide into the non-toxic products, water and oxygen. And then name 2 organisms that test negative for this enzyme. So remember to answer 3B. It's got the semicolon there. I guess it should be a question mark, and then you'd see the question. 4, which rapid test is used to differentiate between the following? So what enzyme do we use to distinguish Staphylococcus species from Streptococcus species? What enzyme do we use to separate Staphylococcus aureus from other members of Staphylococcus? And then what enzyme do we use to separate Enterobacteriaceae, such as E. coli, from the Pseudomonas? 5.
What enzyme or what test?
It's both an enzyme and a test. So the test is named after the enzyme. Okay. Any question about that? A 5, concerning specimen collection for a routine throat culture, what areas of the throat should be swabbed? That's A. B, what areas of the mouth should be avoided? 6, what is the most common cause of bacterial pharyngitis? If you don't know what pharyngitis is, look it up. Okay, gitis is just inflammation, and you can look that up. You must name a species. You cannot name a family because not all members of the family cause bacterial pharyngitis. You cannot name something like gram-negative infection, and it does ask the question bacterial, so don't bother giving me a viral disease, okay? So name a bacterial species for 6. 7, what test is used by microbiologists to differentiate between beta hemolytic group A Streptococcus, the Streptococcus pyogenes, from other beta hemolytic streptococcal species isolated from the throat? 8, answer the following concerning a child's throat culture. State 2 non-selective agar plates that are inoculated to grow a child's throat culture. 8A1 and 8A2. B, what organism can be isolated on CAP, that's chocolate agar plates, but not BAP, blood agar plates? So name a species. 8C is crossed out because you do not need to answer it. You'd have to do some research on it online. It is not discussed in the lab module, so I've crossed it out. I'm not even sure why the main lab instructor, who's currently in Australia, even put that there, but he did, but I crossed it out. 9, list 3 methods that are used to collect urine for routine culture. 10, list 3 reasons why urine specimens should be rejected for culture. Okay, and that's discussed in the lab module. You can read about it. 11, how much urine is placed on the surface of a blood agar plate? 12, what is the most common bacteria isolated from patients with cystitis? If you don't know what cystitis is, I'll actually tell you that one. You can look it up, but it's a urinary tract infection. 13, identify the Gram reaction and the cell morphology. Morphology is shape, not the growth arrangement of these bacteria. The Gram reaction is whether the bacteria are Gram positive or Gram negative. The shape will be whether they're cocci, bacilli, spirilla, or coccobacillus. Okay. So A, Staphylococcus cephrophyticus, B, Escherichia coli, C, Pseudomonas aeruginosa, D, Streptococcus pyogenes, and E, Haemophilus influenzae. And this one can be categorized as 2 different shapes. And as long as you get one of the 2 shapes, you're correct. Any questions about any of that? All right. This is a fairly simple lab because there is no case study. So go ahead and get started on it. I will be here until 8:30 to answer questions. Please use me if you have a question. If there are no questions and everybody logs out, I'm going to log out early also. So if you have a question, make sure you log in before 8:30, and earlier the better. And like I said, I know some of you are getting together to work on the lab. It's not a good idea, but you are not penalized. All right, any questions?
Yeah, I have a question.
Uh-huh.
So one more time, are you gonna send out in an email the results to the unknown bacteria?
Yes, very late today because the lab is getting my priority. Lab
Okay.
six. Okay. Yes, when the question is asking, is the bacteria bactericidal or is it bacteriostatic? And there is a zone of inhibition. By simply looking, you cannot tell.
Right. So my answer was you can't determine whether an antibiotic is bactericidal or bacteriostatic based simply on observation of the zone of inhibition.
Okay. And is that all your answer is or what?
That was all that I put down.
What
And
did I
then
mark? Because I'm sure I didn't accept that, but what did I say?
you said, okay, then how would you determine it?
Oh, yeah. How do you determine that it's bacteriostatic or bactericidal then? So you're saying that it's not, and I've already said it's not.
Right.
You can't tell. So how do you determine it? You can't look at a cell and say, is that a dead cell or a live cell? Because some people have said, let's look at it under a microscope, and you can't determine that either. So what could you do to determine whether the cell is living?
Can you
dead?
streak from the zone of inhibition onto a new plate and then try to culture it?
Okay,
And
I'm
then
gonna
if it-
start recording here and